Okay, in this installment we're going to look more specifically at organelles, the most common ones that we find in eukaryotic cells. And we'll start first with the obvious one, which is the nucleus. It's the largest organelle in eukaryotic cells. And it's a complicated structure in that it itself has a membrane surrounding it. And that is called the nuclear envelope or nuclear membrane. It's filled inside with this jelly-like matrix called a nucleoplasm. There is a dark, dense structure that can be seen in the nucleus, and that is called the nucleolus. And then finally, what you can't see here is chromatin. Chromatin is formally defined as the complex of DNA and proteins. So when we say that DNA is located in the nucleus, sometimes we'll call it chromatin to reflect the fact that it's DNA not by itself, but it has proteins attached to it. Let's talk a little bit more about the structure of the nucleus. The nuclear envelope that surrounds it is made of two outer membranes, and you can kind of see this down here, that they're folded on top of each other. If we think of this top part up here where my cursor is as the outside of the nucleus and the bluish area here as the inside of the nucleus, we can see that there are these holes, or so-called pores, nuclear pores, that form these holes that go all the way through. And so material can exit and enter, go in and out of the nucleus through these nuclear pores. And that's important because there are some processes that occur inside the nucleus, transcription being one of them, and the RNA has to actually leave the nucleus and go out to the cytoplasm, and it leaves through these nuclear pores. This is another picture of the pores of the nuclear envelope or nuclear membrane, and you can see how they're kind of scattered here. This is what's called a freeze fracture picture of an actual cell and the nuclear pores. Okay, the nucleolus is that dark area that we saw in the first picture. It's a dense mass of material in the nucleus, and some cells have more than one. We know that it's a cluster of DNA and proteins. It's not the DNA that we typically think of as nuclear DNA or the genome. The critical part for the nucleolus is that the ribosomes are actually made here. And so the ribosomal subunits, the ribosome is made in parts, is made here in the nucleolus, but then it has to get out of the nucleus to the cytoplasm. And so these ribosomal subunits leave the nucleus through or via those nuclear pores to get into the cytoplasm. So that, we think, is the main function of the nucleolus, but we really don't know, actually. This is a structure that's not that well understood. All right, so that's a very quick overview of the nucleus. We're going to move now to the internal membrane system, or the endomembrane system, which is made up of mostly the endoplasmic reticulum and the Golgi. Sometimes this is called Golgi apparatus or Golgi bodies. We'll also briefly mention two other structures, the lysosome and the peroxisome, that are associated with the endomembrane system. And this is not a structure like the nucleus that has a sort of defined outside-inside. It's a network of membranes that runs through the cytoplasm of the eukaryotic cell. We'll look first at the endoplasmic reticulum. Sometimes you'll hear people call this the ER. In animal cells, the endoplasmic reticulum is continuous to the nuclear membrane. It extends through the cytoplasm, and we can define two regions, the rough endoplasmic reticulum and the smooth endoplasmic reticulum. They have slightly different functions. In this slide, we're going to talk about the rough ER, the rough endoplasmic reticulum. And this picture gives you an idea of what we think it looks like, these very flattened layers of membrane. The ribosomes that you see dots here, those are ribosomes that give it the rough appearance, and that's where it got its name. The rough ER is thought to modify proteins. There are some proteins that actually go inside the internal part of the rough ER and are chemically modified. And cells that specialize in secreting proteins. To secrete a protein means to dump it outside the cell. Cells that specialize in secreting proteins have a lot of rough ER. So rough ER is about processing proteins. Some proteins, not all. In contrast, the smooth ER is a set of tubules that have no ribosomes. That's why we consider it to be smooth. We think the main function of the smooth ER is for lipid processing because lipids are made inside the tubules. The smooth ER also seems to have a detoxification process. In liver, the smooth ER inactivates waste, certain drug products, etc. The Golgi apparatus is right next to the ER, and we'll see at one point a picture of the whole cell where I can show you this. But in this picture here, we can imagine at the bottom where my cursor is going back and forth, that's where the endoplasmic reticulum would be. And so right next to it is the Golgi. And the Golgi is divided into 3 regions. We have the cis, the medial, and the trans. Proteins and lipids that come from the rough ER and smooth ER respectively enter into the Golgi where they're modified even more. And you can think of the Golgi, especially the medial and trans, as kind of a Federal Express or shipping center because the material that goes through the Golgi is packaged in these little membrane compartments called vesicles. And this is where they're packaged and they end up going wherever they're supposed to go. If the protein is destined to be dumped outside of the cell, it'll travel to the cell membrane where it can be dumped outside. All right, while we tend to focus on the ER and the Golgi, there are other components of the endomembrane system that are very, very important, and they are functionally connected with the ER and Golgi. Lysosomes are probably the one that you hear about the most. They are used for eliminating waste products, misfolded proteins. We can think of these almost as a cell's trash can. The inside of a lysosome is very acidic, whereas the inside of the cell would be probably around pH 7. It's at pH 5. And the lysosomal enzymes that can work at this very low pH, highly acidic environment break down materials for recycling or disposal. Endosomes are another compartment that also have an acidic interior, not quite as acidic as a lysosome. You learn more about this in upper level courses. They're used for protein sorting. And then finally, peroxisomes are a class of internal structures, membrane-bound structures, that help rid the cell of free radicals. So lysosomes, endosomes, peroxisomes, all important and all sort of functionally connected to the ER and the Golgi. Okay, in this picture here, this is an animal eukaryotic cell, and if the nucleus is here, it shows how we can consider one view, that the ER is right next to the nucleus, and then the Golgi kind of extends from the ER. So we said most cells, well, all eukaryotic cells have to have a nucleus. Most have this internal membrane system. I can't think of one that doesn't. We're now going to talk about a structure, an organelle, that most eukaryotic cells have as well, and that is the mitochondria. This is the ATP-producing organelle. There are many mitochondria within the cell. I think sometimes you think that there's just one. There's many, many mitochondria. Mitochondria are enclosed by a double membrane. We'll talk about that on the next slide. And they are the site for respiration, and in particular, aerobic respiration. We will talk a lot more about the mitochondria in terms of what it does when we do respiration. This is the structure of the mitochondria. You can see here an outer membrane that surrounds the mitochondria and faces the cytoplasm. The inner membrane has these folds. These folds are called cristae, and they fold back and forth on each other. And that makes compartments. We have the very inside compartment, And we have this space between the outer and the inner membrane, another compartment. And we'll name those compartments when we get to the respiration section. But the ATP-making machinery is embedded on these folds of the inner mitochondrial membrane.